Everybody. Welcome to Virtual Second Look. We're so delighted to have you here. I hope you've been enjoying some of the previous sessions we've had. I'm going to go right into our lightning rounds, and this is a real joy for me because we really get to expose you to some of the mentors, collaborators, and inspirations ahead of you. I have to tell you, I am so delighted with the lineup of people we have. Our first speaker is Dr. Lorelei Thornburg, professor of obstetrics and gynecology, maternal and fetal medicine. I have to tell you, I first met Dr. Thornburg when we had a crash C-section on a coding patient in the emergency department. We navigated that together. Her composure and leadership stunned me. And apparently I'm on a long list of many admirers because the students very much wanted her to present today. Go ahead, Dr. Thornburg.
All right, so I'm going to talk about how everybody gets to the planet, right? So I'm going to talk about how we split the atom and make one person into two, and how we kind of, navigate the birth process. And, the birth process is one of those things where it kind of is a kind of a one-off in med school, right? It's kind of its own thing. So I'm going to talk about the cardinal movements of labor, and hopefully you can see my slides here, and hopefully you can see me too, because I'm going to do both kind of things together. So the cardinal movements of labor are how people navigate the pelvis to get out, okay? And the joke is that it's kind of baby's first dance party. So why do these matter, right? It's not just a list of things you're learning to just how do people move through the pelvis. It's actually helps us predict if labor's going well. It also helps us diagnose and treat disorders of labor or labor management. It is key to operative vaginal delivery, like forceps and vacuums, because if you don't know how the baby's supposed to move, then you can't pull it or push it the right way to make it do that final curve. So these are the cardinal movements of labor. And if you've ever taken a board exam or a shelf exam, and my daughter's in the background because there's going to be an interpretive dance portion of this, so she keeps popping in and out. But if you've ever taken an exam, they love to put these in order and say, are these in order? Which answer is the cardinal movements in order? So the baby engages, it flexes, descends, internally rotates, extends, externally rotates, also called restitution, and then it expulses. And I'm going to give you a little mnemonic of how to remember this. And I'm also going to show you the little dance of it. And hopefully this will make it very clear and you'll never forget it again. So this is what it looks like when the baby moves through the pelvis. So first, the head engages in the pelvis. And I've got my handy bony pelvis, not yet, and my baby. So first, the baby engages in the pelvis. Then it flexes its head, which means bends its chin to its chest, descends further into the pelvis. Then it internally rotates to put its face back by the mom's butt, so back out of the sacrum. Then it extends to come around the pubic symphysis. It externally rotates to move through the rest of the pelvis, and then it comes on out. So that's the cardinal movements of labor. And if we think about the shape of the pelvis, that's why it does it. So I'm going to do that again when I turn my share off so you can see that better. Okay, so how are we going to remember these? Well, first, let's see them in real time. Okay, so hopefully I did this right. You guys will be able to watch this lovely video. So here we got the baby descending into the pelvis, and then we have it internally rotating to put its face back by the sacrum. So it's going to internally rotate here. Turn, baby, turn, it can make it. There we go. It internally rotates. Then it extends its head to come around the pubic symphysis.
Hey,
and then
I don't think your video is playing.
Oh no, it's playing for me.
It's not playing for us?
Oh no, hold on.
Maybe you want to do...
Let me try this. Can you see it now?
Yes, we can.
Okay. All right, so hold on, we'll back up. Our baby goes back up the birth canal. Here we go. So here our baby engages in the pelvis. It descends into the pelvis. It internally rotates to put its face back by the sacrum to make that first turn to get through the shape of the pelvis. Then it extends its head to get around the pubic symphysis. And then it externally rotates to get that shoulder under the pubic symphysis. And then it expulses out into the world. Okay? So, hold on. It's very classy in here because we have this Lovely music playing in the background. Hold on. If I don't stop it, it'll go to like, some ad for something that we all don't want to hear. Okay, so that's the cardinal movements of labor. So let's now explain those in interpretive dance so that you're never, ever going to forget them again. Okay, so here we go. So if you're not on big screen, you should be on big screen because here's my daughter and I, and we are going to do the interpretive dance of the cardinal movements of labor. Okay, so first you get engaged. Okay, and then what do you do next? Well, next you flex your head and descend the church stairs after your wedding, right? So you flex and descend. Then you internally rotate to kiss your steady sweetie. Then what do you do next? You extend your hand to wave to all the people who've come to your wedding. And then you expulse out into the reception and all the fun things that happen afterwards. Okay, so let's do it again. So you get engaged, right? Then you descend the church stairs, you internally rotate, then you extend to wave to all the people, and then you expulse out into the reception. Okay, so why is this important? Well, it's important because of the pelvis. The pelvis is round at this side and it's round at this side, but this side is widest right to left and this side is widest front to back. So the baby has to go in this way, then it has to make a 90-degree turn to then come around this way. And if it doesn't do that descent, internal rotation, and extension, it won't come around the corner and be born correctly. So knowing the cardinal movements and the shape of the pelvis, it all makes sense together. And it really makes sense when you start to think about operative delivery. So here my lovely assistant is going to hold the baby in the pelvis. And I've got a pair of forceps here, which we still do in the Northeast. And we're going to put these on this little baby's head. And you can see this little baby is tiny, tiny, tiny. So I really should be using some preemie forceps, but I don't have any. So there you go. So this baby is now needs to make this extension, right? And that's what you're doing with the forceps. So the move of the forceps is not just a pull. What it is, hopefully I'm going to get you a little closer here. This way. I'm going to get a little closer. And it's not just a pull, it's an extend. That's why they're shaped the way they are, because you bring the baby down and you extend and you finish that last cardinal movement of labor. And that's why knowing the cardinal movements of labor and the shape of the pelvis will help you be able to predict labor disorders, treat labor disorders, and figure out why a mom may or may not be progressing in labor. So that is the cardinal movements of labor. The introduction to OB, if you come to the University of Rochester, we spend 5 hours talking about obstetrics, you and me, and we'll do all sorts of things, including kind of some of the history of obstetrics, why obstetrics is historically marginalized specialty. We'll talk a lot about the inherent challenges in a specialty that focuses on a single gender of patients and some of the unique aspects of obstetrical care. So thank you so much for letting me be at your lightning round. That's my time, as they say in the comedy. So
Dr. Thornburg, that is fantastic. You always remind me of exactly why I love working with you. You should see Dr. Thornburg with labors that don't go so well. Her enactions of those are remarkable. All right, thank you. Our next speaker is Dr. Ray Dorsey, MD, MBA. He's the David Levy Professor of Neurology, the Director of the Center for Health and Technology. So what's really interesting about Dr. Dorsey is that his work in Parkinson's disease is phenomenal, but he's also this really interesting person in the intersection between health, technology, and finance. He provides so many interesting lectures to our faculty to think outside of the box. And that's something that I think we at Rochester really prize, and we really appreciate his time here with us today.
Thanks very much, Flavia. First of all, congratulations, rock on. You guys have all been accepted to medical school, presumably have lots of great choices. I'm gonna try to entice you to think about the University of Rochester. Can you see me okay, Flavia?
Sure can, Ray, you look great.
Outstanding. So medical school in 5 minutes or less. Every society creates its own diseases. I'll say it again. every society creates its own diseases. I didn't make that notion up. This guy, Rene Dubos, a French-American scientist in 1959, wrote this really cool book called The Mirage of Health. And he said every civilization has its own kind of pestilence and can control it only by reforming itself. You can think about COVID-19, you can think that, to what extent did we create COVID-19 ourselves? So, poster child for man-made diseases is lung cancer. So I want you to look at this graph and look at lung cancer deaths. This is the United States. So
Ray, you're not sharing just yet.
Hold on. Let's get going. So much for my technology background.
Confidence in you.
All right. Good.
All good.
So I want you to look at this graph. This is graphs of lung cancer. United States, early 1900s. Lung cancer almost did not exist. I'll say that again. Lung cancer, 1900s, United States, almost did not exist. It was so rare that when people got diagnosed with a case of lung cancer, all the medical students and all the attendings would gather around because they never thought they would see another case. Unfortunately, that didn't stay that way, and people started smoking cigarettes, first in England and the United States in the early 1900s, and you saw a corresponding rise in lung cancer deaths 25 years after smoking took hold in the United States, and then 25 years after smoking peaked, you can see a corresponding decline in the rates of lung cancer. And this isn't the only man-made disease. Car accidents, which are probably the leading cause of death for everyone that's watching right now, are clearly a man-made disease. Homicide, man-made diseases. A lot of heart disease is man-made. Now I'm going to talk to you about another disease that's man-made. Maybe. So the disease I care a lot about is Parkinson's disease. So Parkinson's disease was the first rare descriptions in ancient texts, but the seminal description of Parkinson's disease is Dr. James Parkinson in 1817. The guy, 61 years old, a radical. He actually was arrested for a plot to kill King George III. But his most famous is for writing this essay on the shaking palsy. So he's 61 years old, and he's describing a condition that, in his words, had not been classified in the medical literature. And what's taking a 61-year-old guy to make him think about writing about a new disease is because he's observing something new on the streets of London. And he's observing people with a flexed posture, a stooped torso, and a pill-rolling tremor and a shuffling gait. And what's going on in London at 1817? Well, those of you who are good history gurus know that the Industrial Revolution, the height of the Industrial Revolution is 1817. The capital of the Industrial Revolution is London. And the London fog, which some of you may know, had not so much to do with weather and everything to do with air pollution. If you look at the picture, you couldn't even see across the street. You needed to have little boys carry torches so that you could see, walk across the streets. And the air pollution was so toxic that you can see people were covering their mouths to prevent inhaling it. And this same thing, let's see the next slide. Since that description of Dr. Parkinson's disease went from a very rare disease to now the world's fastest growing brain disease, even faster than Alzheimer's disease is Parkinson's disease. Right now, in the last 25 years, the number of people with Parkinson's disease worldwide has more than doubled. And if we don't change, if your generation doesn't lead us to better pastures, the number of people with Parkinson's disease is going to double again. And so why is that? And there are numerous products and byproducts of the Industrial Revolution, including air pollution and pesticides and another industrial chemical called trichloroethylene, which I won't discuss today, that have been linked to Parkinson's disease. And if you look at this graph, this is a graph of air pollution. Sulfur dioxide is produced from the burning of coal and oil. And it's probably not the causative thing, but just a marker for air pollution. So Parkinson's disease first began in Europe, then it came into North America, and then it's been rising most rapidly in Asia, and it's least common in sub-Saharan Africa. And that actually mirrors almost exactly air pollution. First began in Europe, in North America, rising rapidly in Asia. That picture of the London fog should make people think of Beijing today, and is relatively rare in sub-Saharan Africa. But that's not the only environmental factor that's been linked to Parkinson's disease. Synthetic pesticides, man-made pesticides, and actually some naturally occurring pesticides, which are nerve toxins, target the mitochondria that we know are damaged in Parkinson's disease, have also been linked to Parkinson's disease. Quick aside, Parkinson's disease isn't primarily a brain disorder for most people. It actually begins in the nose with loss of smell and the gut with constipation. So it even gives us clues to where Parkinson's disease is beginning, in the nose and in the gut. Two of the biggest environmental factors, air pollution and pesticides, enter through the nose and the gut. So here's one pesticide. It's called paraquat. 20 plus studies have linked paraquat to Parkinson's disease. It's considered the most toxic herbicide ever created. It kills the weeds that Roundup doesn't. When given to animals by Dr. Deborah Koresh-Slechta here at the University of Rochester, it reproduces the pathological features of Parkinson's disease. And it's so toxic that 32 countries, including China, have banned paraquat, but the United States has not. And in fact, in October of last year, the Environmental Protection Agency reauthorized the use of paraquat. So this is a picture of paraquat, and it's used throughout the United States, including up here in Rochester and Western New York, and probably where most of you are living as well. The EPA's own website says that one sip can kill, and we're working hard to try to ban paraquat. So that is all society creates its own diseases. And if you get fired up like I am, you can go to our website at endingpd.org and you can get this postcard. We'll mail it to you, and you can mail a postcard to the White House. So in 1936, ordinary Americans like you and me mailed in dimes to the White House to raise money to create a vaccine for polio. That was known as the March of Dimes. So successful that they raised $2.7 million and enabled Jonas Salk and then later Albert Sabin to develop vaccines for polio. And now there's no need for polio treatments because polio has been eradicated from the United States and from the vast majority of the world. We're trying to do the same thing, eradicate Parkinson's disease by preventing people from ever getting the disease in the first place. So if you think about society creating its own diseases, to the extent that society can create its own diseases, society and humans can help end these diseases. If you want to join us on that effort, go to endingpd.org. And thanks very much. And congratulations to all of you.
Thanks so much, Dr. Dorsey. We really appreciate your time. Nice U of R zip hoodie. I like that one. All right. Our next speaker is Dr. Margie Shaw. She is an associate professor in the Department of Medical Humanities and Bioethics. She uses her law degree, her master's in philosophy, and her PhD in education in the intersection of bioethics, clinical care, and the education of the professional identity of our students. When we talk about biopsychosocial medicine in the University of Rochester, and we talk about the double helix curriculum intersecting science and humanity, Dr. Shaw leaps to the front of our minds in representing that. Go ahead, Dr. Shaw.
Oh, you're muted.
Okay, that's ridiculous. So every one of us has had a Zoom mistake. So welcome to the crowd. Dr. Nobe, thanks for that introduction. I'm going to talk just a little bit about why we stand out at the University of Rochester around medical humanities and bioethics. And I'm going to start by pointing to Dr. Thornburg and Dr. Dorsey, who just used material from the humanities, from history, from art, from dance to help teach. And so that's in fact what we do. Here at this institution, we teach by, can you see that, Slavia, the next slide?
Yep.
You
Okay.
sure can.
Thanks. We teach by using about the human context of medicine by applying humanities-based knowledge and approaches to issues in healthcare. So we use interpretive dance to teach you how babies are born, right? And we realize that it's more than just scientific knowledge in terms of how we need to cultivate an emphatic empathic, and ethically driven healthcare rooted in the biopsychosocial principles. It's these principles that are so important. This is George Engel on the far right and Catherine Montgomery on the left, who started the Division of Medical Humanities and Bioethics here. I want to really point out George Engel's graph in the middle. It's not used, it's depicted this way, but he did not envision it as a hierarchy. But you heard Dr. Dorsey talk about what's above the person in the society, nation, in the biosphere that affects our health. And so we start with understanding that our health and our well-being and our working with patients, it affects both from the range from the subatomic particles all the way up to the biosphere. And as healthcare providers, how we have to attend to the whole range of what it means to be human. And so we incorporate the visual arts, narrative, theater, and all of the teaching that we do. I happen to teach bioethics, and so I rely on Aristotle's general moral theory, which talks about how you see the world will lead you to the solutions that you come to. And that is, in fact, part of our moral response. And so being a physician is a moral endeavor. How does one do that? And how one sees the world as part of that moral response. We use the personal, the professional, the practice framework. So we talk about give you space to think about your own morals and the morals of your classmates and the morals of others, including your future patients. And what are those beliefs and values, and how do those biases impact the way you frame the world? And we talk about the professional obligations and the morals of the profession and how they have changed over time. And we do this all in the context of practice. It's all case-based. And so the reflector is we start with your first class. We started orientation in your first class to support you in your learning. The reflector is a document that was created by one of our almost graduates when she was after she finished first year medical school that helps support learning in anatomy lab. And the document on the right is a growth of that in how helping learning pathology with respect to linking back to what we did in anatomy lab. And the Division of Medical Humanities and Bioethics supports all of that. I want to share another example of how we do this. This one is, so we do this work all the way from first year in the medical school through residency. This is the first year resident's story during an ethics conversation.
We had a patient in the ICU, a 91-year-old woman with profound dementia, a massive arachnoid cyst instead of a right frontal lobe, and failing organs. She could not tell us what she wanted, but her daughter spoke for her. She could not eat. Her daughter requested tube feeds, then a peg. She could not breathe. Her daughter requested intubation, then a trach. Her kidneys failed. Her daughter requested dialysis, then offered to donate a kidney. Our patient is still alive in the ICU.
So what we attend to at the University of Rochester is not just the science that you will need to know to practice medicine, but we attend to what it means to practice medicine in relationships with others, with your patients families, with your colleagues, and how to do that in a way that allows you to become and flourish to be the human and the professional that you want to be. And so we offer all kinds of support to think about how you might respond if you were involved in this patient's care, how you might respond yourself, and how you might respond with your colleagues, and how you might respond to this patient's loved one. And so we will support all the learning that you do at this institution. And I hope you come because I think we're pretty special.
Thank you, Dr. Shaw. That was lovely. I really liked your 55-word stories. We have a number of these posted throughout our admissions area, and they're so impactful. And watching the development of that over the course of the past couple of years has been really powerful. Our next speaker is Dr. Rob Dirksen. He is the Lewis Pratt Ross Professor of Pharmacology and Physiology. He is heavily involved in the teaching of electrophysiology of the cardiac muscles and approaching the EKG to many of our students. He is beloved not only for his mentorship, but his clear thinking and the fact that he is so approachable. He takes complicated issues and distills them into simple principles. And we are really, really excited how much influence he's had on the past and future generations to come. Thanks, Dr. Dirksen.
Thank you, Flavia. I am not muted, so that's nice. It's a change. Want to welcome everybody to this day and introduce myself. I'm professor and chair of pharmacology and physiology. We're on a research program focused on developing new treatments for muscular dystrophy. And for the past 20 years or so, I've been teaching nerve muscle physiology and human structure function, which is the first course that our medical school students take in the fall, as you know. Today, I'm going to be giving you a lightning lecture on how we use the EKG or the electrocardiogram to reinforce fundamental principles of cardiac physiology. And I'm just going to go ahead and share my presentation. How we use the EKG to reinforce these fundamental principles of cardiac physiology and how exercise can alter the electrical activity of the heart. So we have to first talk about the flow of electrical activity in the heart, and that here's our heart here, and the normal flow of electrical activity starts in the SA node. This collection of cells in the upper right-hand part of the atrium that has this automaticity, ability to generate their own action potentials. And the function of that is to set the heart rate. As those action potentials permeate and extend into the adjacent atrial muscle cells, the atrial muscle cells become activated to initiate action potentials that propagate through the atrial muscle. And of course, that triggers the atrial muscle to contract and to squirt the blood from the atrium into the ventricle, which the ventricle will need in order to feed blood to the rest of the body. As the electrical signals progress to the AV node, the atrioventricular node, which is located right between the atria and the ventricle, the job of the AV node, the properties of those cells are to slow the propagation. It's kind of like a speed bump in the highway. It slows the propagation of the action potentials to the ventricle, and that gives an important delay so that the atria contract first and give the ventricles enough time to fill with blood to expand, so that when they need to contract, there will be a full load of blood to expel to the body. So once the action potential leave the AV node, they activate action potentials in the Purkinje fibers, which is a conductive system that rapidly and uniformly delivers those action potentials to the entire endocardium of the ventricular wall. And then the action potentials spread across the ventricular wall from endocardium to epicardium through these depolarizations to trigger myocardial contraction. And obviously now the blood has been put into the ventricle, and the contraction of the ventricle will expel the blood and deliver it to the rest of the body. So that's the general electrical flow that happens, but the EKG is a very simple and cost-effective, non-invasive approach to monitor the electrical activity in the heart during the cardiac cycle. Surface electrodes are placed on the individual. This is a 12-lead EKG, which is standard in the clinic. The lead 2 EKG is what we focus on in class because it is the most widely used EKG vector, and it consists of placing the negative electrode up here on the upper left and the positive electrode on the upper right and the positive electrode on the lower left. And with this orientation, as the electrical activity goes from SA node through the heart, through the ventricles, there's this propagation wave from the top to the bottom towards the positive electrode. And that creates these positive signals on the lead 2 EKG, shown as the P, QRS, and T wave. And they reflect these fundamental activities of the physiology of the heart, meaning the P wave is that wave of depolarization that goes through the atrial muscle. The QRS complex is even larger because the ventricle is larger. That's the wave of depolarization from endocardium to epicardium across the ventricular wall. And then you get the repolarization of the T, is the T wave. And the intervals are very important too. The PR interval is set by that delay of the AV node to allow for the time between atrial and ventricular contraction. And the time between the QT interval is really when the ventricles are depolarizing and repolarizing. So it gives us an indication of the action potential duration. Now, the overall EKG waveform is relatively consistent from individual in normal healthy individuals, but the duration of these different events can vary across age, sex, race, and disease state. For example, for a given heart rate, the QT interval here is much longer on average in women than men of the same age. And that's an important variable because long QT can predispose people for an arrhythmia. So it is an important parameter to be watching in women. The other thing is that even in a given individual, the EKG parameters are not static. They're changing all the time in response to sympathetic and parasympathetic stimulation to the heart. And to help illustrate this point, I've recruited a colleague of mine to help show how the EKG can change. Come on in, David. During exercise. So let me introduce you to my colleague, David Ewell. He is a professor of pharmacology. He's also the co-director of human structure function, and he's my boss in that course. And so I appreciate that he came in here. As you can tell, he's also an exceptionally well-trained athlete. And so we're going to take a look at his baseline EKG. I can show you that in a second. Hold on. Are you able to see this, Trace?
Not yet.
Yep. Well, he hasn't flatlined. So let me start.
It looks like he's still breathing.
He's there. All right. So this is his baseline EKG trace. I'm going to stop it here. You can see, obviously things are, this is a live trace. We're going to see things are a little bit wavy, but you can see
I can't see anything just yet, Bob.
Oh, you cannot.
You're not sharing.
Sorry.
That's okay.
Here we go. Can you see this now?
Yes, we sure can.
Okay. I'm going to stop here and go backwards a little bit to where we had the trace. Okay, so here you can see the P wave here, the QRS complex, and the T wave. So now I have David do some exercise.
All right.
So we're going to have him do a little bit of exercise to try to get his heart rate up. So you can see he's doing push-ups. Pretty soon he's going to go to a one-arm push-up. Just want to get his heart rate up. Want to increase it. All right. So let's see if we have now. New rhythm. And what you can see is that if the distance between these QRS complexes are much shorter in duration compared to back here, they're much longer. Also, what you'll see is the time between the QRS and the T wave, that QT interval is much shorter as well at the higher heart rate. So you can see this is much longer in before the exercise. And harder to see is this interval between the P wave and the QRS complex. That also gets shorter with exercise, this interval here between the P wave and the QRS. And I'm going to try to show that a little bit more clearly in the next slide, because that's always a harder one to see so let's go back to the presentation. Right. So here I have a resting heart rate, and you can see underlining the long distance between the QRS complexes compared to after exercise. I think you can also see the QT interval here is much shorter than the QT interval under the control condition, and harder to see, but even the PQ interval is also reduced. So in the course in HSF, we actually study the fundamental mechanisms by which all of these events change under autonomic regulation. So the last thing I'd like to just say is thank you all for the opportunity to present this, and thank you to Dr. Ewell. You'll obviously get to know him very well in the fall as your course director of HSF, and I appreciate your coming by. And we all hope to see you here in August when the course starts on the 16th.
Thank you so much. And I just want to tell you, I'm very impressed, Dr. Ewell. And Bob, if you ever want to have anybody in SVT, you can ask me to do that. I'd be happy to be a monitor for you right there.
That would be great.
Yeah. Thank you. Our next speaker is Dr. Sarah McConnell. She is an assistant professor of neurobiology and anatomy, again, from that EPIC course, Human Structure and Function, which is the very first course you take coming into medical school. Dr. McConnell is really beloved. She is a faculty member that so many students go to for mentorship, for guidance, reassurance, but also as importantly for knowledge. So thanks, Dr. McConnell.
Hey, thank you so much, Dr. Nove. Appreciate your introduction. And good afternoon, all. Thank you so much for joining us. Indeed, I teach in HSF, Human Structure and Function, and there you would see me in the anatomy labs, the histology labs, as a PBL tutor, lecturing on anatomy and histology, and also, if I can remember to share my screen over here, this one looks promising. Hopefully you can all see a slide that says early embryonic development.
Yep.
Heads nodding. Awesome. Thank you. So this is the other topic that I lecture on, early embryonic development. And so I thought I'd give everybody kind of a taste of this, since not many people have much background in embryology, and it's seriously the most fascinating stuff. So I'd like to introduce something cool about each of the first 3 weeks of your existence in utero. So starting at the very beginning, this whole thing starts with fertilization. When the egg cell meets up with the sperm cell, they fuse, they combine their genetic contents, and now you have a zygote shown over here. We're looking at kind of a cross section through the uterine tube, a little bit of the uterus over here. This process of fertilization happens within the uterine tube just shortly after ovulation. I think this is so much cooler than a birthday. I mean, this is existence day. Like, this zygote, this genetically unique individual, did not exist prior to this event. So congratulations, you exist. Now, it's like in Jaws when they say we need a bigger boat. We're going to need more cells. One cell is not enough. So mitosis, undergo lots of rounds of mitosis. Replicate DNA, divide. Replicate DNA, divide. One cell becomes 2, becomes 4, 8. You know the math from there. So you've got kind of a big old glob of cells that then needs to get more organized into a structure that we call a blastocyst over here. I really care about 2 parts of this blastocyst. There's the inner cell mass that's shown in blue. Being a narcissist, we like that part best because those are the cells that will form the entire embryo, that will form the entire you. Like, all of your cells ultimately can be traced back to this little glob of cells. I also care about the cells in green out here. These are called the trophoblast. Tropho comes from, pretty sure it's Greek word for to nourish or to feed. And so the trophoblast will help form the placenta, that interface between the embryo and the mother, your supply of nutrients and oxygen, this nourishment. Okay, so we're getting organized, and meanwhile, we're moving along the oviduct, being propelled by muscular contractions along this tube, and on the surface of the cells that line this tube, there are these little cilia, these little tail-like processes that kind of beat the blastocyst along in the correct direction. So it goes toward the inside of the uterus over here. And about the end of this first week, that blastocyst will find a nice spot on the inner lining of the uterus, grab hold, and implant itself. So I've got a 5-second animation to show this implantation process with the trophoblast, these green cells grabbing hold of the surface of the inner lining of the uterus, just working its way under that tissue. Did the animation play for all of you?
Yep.
Awesome. It was so short. Let's do it one more time. 5 little seconds. There goes this blastocyst. And you'll notice that the endometrial lining, those surface cells of the uterus, close up behind it. It's really like planting a seed. You know, you make a little hole, put the seed inside, and cover up the earth over it, keep it nice and secure in place. Same thing happens during implantation. So, gosh, what a week, right? We didn't even exist. Now we exist. We've got more cells. And we found a nice spot to implant. So let's cruise on, because there's still a long way to go. In week number 2, it's like the embryo is counting up with you. That inner cell mass, that little glob of cells that looked, it was colored in blue before, it starts getting organized now. It gets organized into 2 cell layers. And I should mention, we're always looking at a cross section through a spherical structure and just kind of chop off part of it and look inside. This is what you would see. Now we have 2 cell layers because it's the second week. One of these is on the more dorsal side. That's this tall layer in blue. One is on the more ventral side. And we don't even know which way is the head at this point. We've got a back side, a dorsal side, and a belly side, a ventral side, and, making progress. Meanwhile, I just want to highlight what's going on with the trophoblast. That's getting more elaborate out here. It's starting to form the placenta. as it invades this uterine tissue, it's forming a network of spaces. These white spaces out here are called lacunae, meaning basically spaces, gaps. And as it continues to invade the tissue, it will start eroding these maternal capillaries, these blood vessels, so that they will bleed into these spaces it's creating, and that will provide the oxygen supply and the nutrients for the developing embryo as it grows. Very conveniently located. But really my favorite week is week number 3 over here, where the embryo now has 3 layers. So it's seriously counting right up with you. So what used to be 2 layers now is 3 layers. Each layer will give rise to different portions of the embryo. Ultimately, we've got the ectoderm. Ecto means outside. So one of the parts that will give rise to is the skin, going all around the outside of you. It also forms the nervous system. The mesoderm, in pink over here, middle layer, will form muscle and bone, and the endoderm, endo meaning inner, will form that inner lining of your digestive tract. So cool, we're a disc with these 3 layers, but we've only got skin kind of on the back side, and we've got this like exposed gut surface on the other side. So now it's time for my interpretive dance. I'll stop sharing. Let's see, you don't see my screen anymore, right? Like you just see me? Awesome. Okay. So I'm going to be the embryonic disc, right? So I've got yellow endoderm, that's my exposed gut surface. After that, I've got my pink mesoderm, that's ultimately going to give rise to muscle and bone. And to complete the disc, here in blue, it's my very most favorite ectoderm shawl. Okay, so I'm a disc. I was once, so are you. And basically what happens is the edges, those lateral sides of the disc, have to bend around and fuse in midline. So that ectoderm fuses to ectoderm, and mesoderm fuses to mesoderm, and endoderm fuses to endoderm, forming now a closed tube. So just this kind of giving yourself a hug, sort of a gesture there. I'll bring back my slide. Share. Make sure I can see all of you again. All right, so, and you can see my slide again, right? Excellent, great success. So that trilaminar disc has now folded up into a tube with basically skin, ectoderm going all around the outside. Now we also have a clear head end and a clear tail end. And from this tube, little limb buds will extend coming out the sides so that you can really start to see how your body plan emerges from this very primitive structure. Then for some foreshadowing, going from here, we've made it from fertilization, forming a zygote, 1 single cell, up to forming a trilaminar disc here at week 3. From weeks 3 through week 8, this is like the most extreme multitasking you've ever done. You lay the foundations for all of your organ systems at once. Nervous system, cardiovascular, respiratory, reproductive, everything. You start building everything at once, so that at the end of week 8, you really kind of look like a small human. And then from there, all of these systems just mature until you need to remind yourself what Dr. Thornburg had told you about those steps for, proceeding through the birth canal and getting a baby out into the world. So these first 3 weeks really count up pretty nicely. With week 1, you start with 1 single cell that gives rise to many cells, gets organized and implants itself. In week 2, you now have 2 layers of cells and you start building that placenta. And in week 3, you have 3 layers of cells, and that disc folds into a tube that ultimately will give rise to you. And at any point, anything can go wrong. So it's kind of a miracle that any of us make it through this whole process. So congratulations to all of you, both for that and for getting accepted here. And I really hope to see all of you in the fall. Thank you for your attention.
Thank you so much, Dr. McConnell. I always say, and I genuinely mean this, I wish we could go back to medical school after we've been a clinician for years and years and years. It would be remarkable. I love that. I absolutely love that. All right. Our next person up is a friend of mine, Dr. Valerie Liu. She's an assistant professor of emergency medicine with a fellowship in global health. She is our institutional champion around issues related to environmentalism in hospital settings and in planetary health. She is a mom of 2, an avid traveler to remote isolations. And when I say remote, I mean remote. Dr. Liu also leads much of our wilderness medicine initiatives for the medical students and the interest groups as well. So I can't see everybody right now, but I know Dr. Liu, you're somewhere on here.
Yes, I'm right here looking a little ragtag. Nice to see these faces, some familiar, some not, some new. It's wonderful. Thank you for that introduction, Dr. Nobe. As Dr. Nobe said, I do things related to climate change and healthcare, and I'm also one of our clinical faculty for emergency medicine. So I thought I'd take this opportunity to share with you guys one of my main interests, which is a concept called planetary health. Who here has heard of planetary health before? All right, Margie Hodges, Flavia Nobe, raise some hands. All right, and I think there was something in the chat too. Yes. All right. So I'm going to use a PowerPoint just to kind of keep me organized and do some visuals. So let me pull that up. But yeah, I love interaction, so feel free to shout out things at me, or if you have a comment or a question you desperately need me to answer, just shout it out. Okay. So yeah, I wanted to talk to you guys about planetary health and how the University of Rochester Medical Center is involved with planetary health, both from a practical application and starting now from a research standpoint as well, for those of you guys who are so inclined. If you have been watching the news cycle at all for the past decade, you've seen images that look like this: wildfires, drought, melting glaciers, extreme weather events. And I'm sure you've also seen this in the news, graphs that look like this related to global temperature. The world is getting to be a hotter place, but it's not getting hotter in a way that's very predictable. So rather than the concept of climate warming, a lot of people have turned onto this concept of climate energizing. So there's an increase of energy going into our planet. So not just warmth, not just heat, but energy that has all sorts of unpredictable effects. Things like jellyfish blooms clogging nuclear power plant engines, things like algae blooms, things that are just not predictable in a just temperature increase way. And an analogy I like to use is that of a toddler who's gotten an influx of sugar, right? So not just clear, predictable outcomes, but all sorts of crazy behavior. And that in essence is what's happening to our planet. And why does that affect us as future healthcare professionals? Well, the climate affects health. So the United States put out the 4th National Climate Assessment, which comes out every 4 years in 2018. And it said basically in no uncertain terms that the health and well-being of Americans is already affected by climate change. So this isn't something that's happening 50 years from now or 100 years from now or happening somewhere else, like if you live in a coastal community. It's happening right here and it's happening right now. And I can't give a talk to future University of Rochester students without bringing in the biopsychosocial model. So if you guys have heard these terms, this is actually a model of medical teaching that originated in the University of Rochester. And it's basically about how it's not just like a molecular pathway that leads to a pathogen that leads to disease, but there's all sorts of social and behavioral contexts that are also now mixing with things happening in the environment, and that results in an exposure pathway that leads to health outcomes. So in order to be a complete physician that really understands the true meaning of disease, how to treat disease and prevent disease, you also have to understand what's happening in the environment and what's happening socially with a patient and how that all interacts to lead to a disease process. So planetary health is really the conglomeration of those things. It is the health of human civilization and the natural systems upon which they depend. So if you hadn't heard about it before, that is planetary health in a nutshell. So it's human health, ecosystem health, and where they intersect. So in terms of disease outcomes that are related to planetary health, it's really almost anything you can think of. If you are going into pulmonology, or cardiology. Asthma and cardiovascular disease are known to be linked to air pollution, which is due to rising temperatures and more extreme weather. If you are going to be an infectious disease doctor, cholera, cryptosporidiosis, Campylobacter, leptospirosis, all those are related to water quality impacts, which are in large part impacted by rising sea levels. If you are interested in metabolic health, because you're going to be a pediatrician or internal medicine doctor, malnutrition, diarrheal disease, even obesity is linked to water and food supply impacts. So in emergency medicine, what I do, we actually see these impacts every day. When there is worsened air quality, or say you're a doctor on the West Coast, or like I was in Indonesia for a while, whenever there's forest fires, increased hot weather warnings, people have worsened asthma and COPD exacerbations. Right here in Rochester, in the last 10 years, there's been an explosion of Lyme disease. Not dengue, not yet, but Lyme disease incidence has gone up a lot in the emergency department. And we have pediatric and elderly populations coming in all the time with heat exhaustion. And these are directly linked to impacts due to climate change, due to climate energizing and planetary health. So what can we do about it? I feel very lucky to be part of an institution where we have lots of linkages to international, national, state, and local agencies that are impacting climate change. So we have partnerships with the Medical Society Consortium on Climate and Health to Healthcare Without Harm, and our CTSI, so the Clinical Translational Science Institute, one of our major research institutions in the hospital and in the University of Rochester, just partnered with the Planetary Health Alliance. And we are trying to find projects and researchers right now who are interested in making an impact on climate and planetary health. On the national and state advocacy level, many of our residencies lobby for change. So the emergency department sends a gathering of faculty members, residents, and occasionally med students as well to participate in Lobby Day, which happens every spring, where we talk about issues important in healthcare to our state representatives. Rochester itself is part of the We're Still In movement, which is the United States stepped out of the Paris Agreement, but lovely Warren, who is our mayor, said that Rochester is still in on reducing carbon emissions and making steps to fight against climate change. Rochester actually has a climate action plan. So if this is something that's really important to you, know that Rochester has been looking really closely at its greenhouse gas emissions and seeing if we can reduce greenhouse gas emissions by 40% by 2030. And they're investing money, they're investing research and lots of personnel in trying to make this happen. And our own University of Rochester Medical Center actually contributed as Voices from Healthcare in our climate change resilience plan to see how we can make our city and our hospital system more robust in the face of climate change. So a really sobering statistic that I like to think about is that the healthcare system as it exists today is actually part of the problem. Healthcare accounts for almost 1/10 of US greenhouse gas emissions. And when you think about how many, what tonnage of greenhouse gas emissions the US is responsible for, it's actually more than most countries. So it weighs heavily on me that the hospital system is contributing to this issue, but it's also a way to make a difference, a way to make a big impact. So things that the emergency department and the University of Rochester is working on related to greenhouse gas emissions from hospitals are manyfold. So we actually started something called the ED Green Team, where we have a group composed of faculty, residents, nursing staff, environmental services, patient care techs to figure out ways to basically attack some of these climate issues and hospital waste issues in a way that also takes into account clinical care, because our first responsibility is first to the patient. So we've done projects related to, actually I accidentally deleted this slide. We've done quality improvement projects working on decreasing inappropriate biohazard waste. We've pioneered using a BioQuell machine, which is a hydrogen peroxide mister, to sanitize hospital supplies that would have otherwise been thrown away, to reclaim them and be able to restock and reuse them in the hospital. And we were so successful in this that infection control actually picked up our project and spread it to the rest of the hospital. And during COVID times, they've used that same BioQuell process to sanitize N95 masks so we could reuse them. So that's been pretty amazing. It's being carried forward too in future years. Our medical students are incredibly active in climate change. So our chapter of Medical Students for a Sustainable Future is working on surveys to improve their education. They're working on research projects, and they came up with this petition to President Mangelsdorf and Dean Taubman to support LEED Platinum certification for our new hospital building that's being built. So there's an incredible amount of activity happening in the medical school. In the larger University of Rochester, we are actually piloting this movement called the Little Book of Green Nudges. And we are one of the pilot institutions that's part of the UN Environment Program to make these little changes happen that add up to have a big impact. And it's actually a concept from public health that previously has been focused on like obesity, diabetes, and smoking cessation, things that are very hard to change. But if you make little nudges, things that basically are easy to accomplish and not too painful, they add up to make a big difference. And they're using that same approach to tackle climate change. And I like to just end this with a little bit of a call to action, right? For those of you who aren't convinced, why us? Why the medical community? I think as physicians, we have a moral imperative to help those who are not in power, who are very susceptible to disease. We are also natural educators. It is basically our job as physicians to translate research and science to the public. We're collaborative, and I think about on any ED shift, I could be talking to 3 or 4 different disciplines, like a cardiologist, a urologist, a trauma surgeon to take care of a patient. And this is a big problem that's going to require a multidisciplinary approach. And finally, we are society's safety net. It all trickles down to us. The bottom of all those social issues, those environmental issues, all come down to that health outcome, which is the patient sitting in front of us. And I have to say, we should be acting on the precautionary principle. We're seeing a lot of these warning signs with human health and environment. And that's kind of like a patient just starting to become tachycardic or just starting to become hypoxic. If we wait until the Earth's vital signs look like this, so extremely tachycardic, hypoxic, hypotensive, tachypnic, this is a patient with all the SIRS criteria, right? This is like a septic patient circling, going down the tubes, basically. If we wait until the Earth looks like this, we are too late, right? As clinicians, we try to catch the problem before it becomes irreversible. And that's how we treat health. And I think we have to think about the same way with climate change and health. So, I think this is an old view of physicians in medicine where, especially for emergency medicine, we're very reactive, right? This is like, patient's leg is bleeding, no time to call surgery, tourniquet and amputate, quick, throw me a scalpel. And it's a very reactive approach. But I think medicine in the future, so you guys, is gonna look more like this. So, really harnessing our ability to be advocates for health, working with national, international organizations to make change. Yeah, and I'm very excited to meet you guys. So.
Thank you, Valerie. I really appreciate it. That was wonderful and definitely a call to action, as you have been absolutely saying for years and years and years. All right, I'm just going to move on to our very last speaker, Dr. Jessica Shand, who's an associate professor of pediatrics and hematology oncology. Additionally, she's an associate professor of bioethics and a phenomenal marathoner. Really, maybe gave Dr. Ewell a little run for his money. We're really lucky to have Dr. Shand here because she's also just newly appointed to be the pathway track director for our medical ethics pathway. So very excited to have you, and thanks for joining us.
Thank you so much, Dr. Nobe. I am going to share my screen as well. It is fantastic to meet you all. This is my first time doing this, and I'm just so, I'm so inspired all the time by my colleagues. So the title of my talk today is called Social Justice is in Our Blood. So I am a hematologist. I love hematology because there's hematology in everything. And as Dr. Shaw might have mentioned to you earlier, there's also ethics and justice in everything. So I'm going to talk to you a little bit about what happens when those two things intersect. So a bit about me relative to the University of Rochester. You should know that the University of Rochester School of Medicine and the medical institution is a place where people with diverse interests, different interfacing interests, interprofessional, interdisciplinary interests thrive at every level. And when your faculty thrives by being interdisciplinary and outside the box thinkers, your students, your trainees, everybody thrives. And that's something just that I cherish every moment as a faculty member and as a clinician at this institution. As Dr. Nobe mentioned, I'm appointed in two departments, pediatrics and medical humanities and bioethics. And you'll see a bit of me through the 4 years of your medical school. I will teach all of your chemotherapy lectures, probably wearing this shirt that says DNA checks itself before it wrecks itself, but it doesn't always. And those 4 hours of lectures are going to tell you exactly how we do that. As was mentioned, I am the newly appointed bioethics pathway director. I'm going to be involved in your bioethics curriculum in year 1 and 2. I also teach medical humanities selectives on the intersection of values and belief and spirituality in medicine. I am, with Dr. Liu and others, one of the Medicine in Motion interest group faculty advisors. So as Dr. Nobe mentioned, I am, they call it the master's division. After you cross that 40-yard line, you get a different, you get bumped up into a different age category. I do compete in the marathon and the 50K. So you'll see me around in some of the wellness spaces where we talk about activity in medicine. Later on in your career, I'm a preceptor in pediatrics and also a member of the clinical ethics consult team. So this is really a place for me with my training in pediatrics, in hematology oncology. I have a background in cellular therapy and immunotherapy, and in other areas, a place to thrive. And I'm so excited to share that with you. A little bit about me. People talk about work-life integration, but I X'd out the work of the work life because it's all life. We bring ourselves into every patient encounter, into every learning experience, and we take the best and sometimes the worst of it, and we integrate it into who we are. And that's probably a good time to tell you that I'm the Department of Pediatrics lead on clinician well-being, but that's another story altogether. My husband is an emergency physician, so I love emergency medicine almost as much as Dr. Liu without actually practicing it, but I think it's the most amazing. The emergency department is just the most amazing place. Most of us clinicians get to choose which patients we see. I love children. In the emergency department, you take care of everybody no matter what, and that's a beautiful, just thing. So social justice is in our blood. And when we think about hematology and we think about research and we think about the global burden of blood disorders, a lot of things come into focus. So some of my areas of interest, how do we manage blood products as a scarce resource? That's going to be particularly important now during the pandemic. We have fewer people coming out to donate blood. Blood products such as platelets actually require multiple donors. So what's going to happen? What's going to happen for Dr. Liu in the ED on the trauma bay side when we tend to need a lot more blood, especially over the summer, and there may be local, national, global shortages of blood? How do we manage that? That's a hematology issue. It's also an ethics and a justice issue. My personal area of interest, how are national and global research priorities set, right? Those of you that may be interested in research, interested in diseases, the way we get research done is by funding, but there are people who decide what gets funded and what doesn't. And so really important questions about how do we decide what research gets funded and what are the implications for developing therapies. And as a pediatrician, right? So as a pediatric oncologist, pediatric childhood cancer research makes up only 4% of the National Cancer Institute budget because kids are young, they don't vote, and thankfully pediatric cancer is less common than adult cancer. But think of all those years of potential life lost when children die of cancer. So who sets those priorities? On the ground level, I'll tell you a bit about how can we improve community health and primary care for diseases like sickle cell disease, which is my background. I'm sitting in front of some of my sickle cells. Hemoglobin disorder, single gene disorder, one of the most common hematologic issues worldwide. And how do we improve primary care screening for things like iron deficiency and nutritional anemias? If you come and rotate with me in pediatric complex care clinic, we'll talk a lot about that. So this intersection of ethics and science and community health, and Rochester does that just so incredibly well. So I'll give you an example. This was an essay published in the Journal of the American Society of Hematology. So this is our hardest hitting academic journal about low white blood cell counts, or what we call neutropenia. And it turns out that individuals from African heritage, also Mediterranean and Latinx heritage, tend to have lower circulating white blood cell counts. And there's a subset of those patients where a particular subset of their white blood cell count, the neutrophil, and the neutrophil is kind of like your strong arm, super oxidative, bacteria fighting cell, are low. And they're low to the point when people screen blood counts, they may look at it and think, gosh, there might be something wrong with that person. Well, what's wrong a lot of the times is just that the normal value range for a long time has not been inclusive and diverse. And actually that subset of people with a lower white blood cell count don't have a condition, don't have a diagnosis. It's just what's normal for them. And so how do we educate about that? How do we talk about the science behind that? And how do we eliminate unnecessary referrals and scaring the pants off of a parent who might think that there's something wrong with their child's blood when it's really just part of who they are? And I'm going to tell you why that phenotype is what it is, because it's really, really cool. Duffy antigens on your red blood cells are good, but they're also bad because they're used by the malaria parasite to invade red blood cells. So it turns out that there is a particular phenotype, blood phenotype, that downregulates the expression of certain Duffy antigens, which then renders the red blood cell resistant to be invaded by malaria, which is awesome. That downregulation of that receptor also has consequences for how white blood cells behave. It doesn't make them work any worse. It actually makes them a little bit better at getting out into your tissues and doing what they do. But that phenotype makes your circulating white blood cell count, particularly those neutrophils, appear lower. But so you can understand why in patients with African and some Mediterranean and Latinx descent who have this phenotype, they have perfectly healthy blood cells. There might just be fewer of them in circulation. They're out there in the tissue. They're really efficient waiting for things to happen. And their red blood cells are resistant to malaria. That's not a problem. It's not a condition. It doesn't need to be referred, and nobody needs to worry about it. But if we don't know the science, then we don't understand. And we're not going to know the science unless the money is spent to do the research to understand it. So that gets you back to the ethics of what research priorities we set and what we consider important. So again, the University of Rochester is a remarkable place to be a part of the intersection of translational science, clinically applied science, clinical medicine, interprofessional medicine, and community health. So when I talk about hematology and social justice, you might ask, well, where in my medical school experience can I witness it? Where is it happening? Our pediatric oncology clinic has a comprehensive sickle cell disease clinic that our Dr. Susie Nerona is a national expert in sickle cell disease. She and I trained together at Johns Hopkins. We're really blessed to have her here. And she does tons of community education on hemoglobin disorders and sickle cell. We're also one of the few places in the country, so we have one of the best combined internal medicine and pediatric residency programs. And we have a remarkable clinic called a complex care clinic, because we recognize that young people between the ages of 16 and 17 and 30, particularly if they have chronic diseases like sickle cell, kind of get lost in that transition between their pediatrician and their internist. So we have a clinic that focuses particularly on those patients. And for patients with chronic anemias and sickle cell disease, this is an awesome place to make sure their primary care is taken care of. When we talk about ethical issues in management of transfusion, we've got a remarkable transfusion medicine department. We've had several students do research with our transfusion medicine experts. And when we have to talk about how we give transfusions as a scarce resource ethically, that involves our clinical ethics consultation service. Another thing you should know about Rochester is the strength of our health and relationships with the community, and particularly with advocacy groups. So I keep mentioning sickle cell disease because sickle cell disease is an important chronic disease in pediatric hematology, primarily affecting children of African, Caribbean, and some Latinx and Indigenous descent. We have an amazing parent advocacy group called the Rock City Sicklers, and our scientists and doctors go to events all the time. Most recently, we went out to talk about the safety of the COVID vaccine and really addressing some of the mistrust around vaccination that some communities have around science. And you have to know your history and you have to know your ethics. And so our students who are interested in these points of integration are welcomed and encouraged to work with us in community health endeavors, in our clinics, and doing the research. So it's just a really exciting place to be at the intersection of science, clinical application, ethics, and community health. And what I've told you about hematology and social justice is just one of the many examples of that I happen to be interested and deeply involved in. And so I thank you so much for your attention. I really hope to see all of you back in a few months in ethics teaching and in other places in your primary clerkship in pediatrics. And I hope you all have a wonderful day. Thank you.
Dr. Sheehan, thank you so much for that really interesting conversation. I really appreciate it. And I believe, if you don't mind, Alicia, telling me what I need to do next, I think we're on a break.
Yep. So we're going to go ahead and break now for the next 20 minutes or so. And our final formal session before tonight's trivia is going to be the class year panel. So that's going to start right around 3:30, and we'll see you all back there.
Briefly, just one more time, if anybody has any questions about the speaker's topics, you can send them to me. I will forward them along. Additionally, one other thing, I really want to thank our speakers. It is Saturday, which we all recognize. I know medicine's a 7-day-a-week gig, but I really do appreciate your time tonight. Bye, everybody. We'll see you later.